This is episode 99 of the Immunology Podcast, Inflammation and Innate Immunity with Dr. Luke O'Neill. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Luke O'Neill from Trinity College Dublin on the podcast to talk about his research on toll-like receptors, innate immunity, and signal transduction and inflammation. We've also got our usual roundup of recent highlights of immunology news coming up, but first...
Immunology 2025 is coming up in May in Hawaii. We'd like to remind everyone to book your hotel soon. They run out, since you can get a discounted rate until March 31st. Book now at immunology2025.ai.org/hotels. Hello, Jason.
Hello.
How's it going?
Oh...
Should we talk about this? Should we just ignore reality and pretend we live in a beautiful world of scientific discoveries alone?
I'm concerned about NIH funding.
Yeah. Man, this is such a bummer. That is not even enough to describe the level of despair I'm feeling at the moment. So I can imagine you and all of our US-based listeners.
It's just I'm waiting for I'm trying to be patient and wait for the storm to pass and see how much of it actually sticks past the lawsuits.
Yeah. Well, now that we have acknowledged reality, let's just talk about fun stuff. There's for those who want to escape reality and listen to more podcasts beside this one, we do have the second season of Lab Coats and Life podcast coming out on March 5th. Our sister cousin podcast with our own Nicole Quinn at the helm. We had really nice conversations about different areas of, the academic, non-academic, scientific journey. What was your highlight of this season?
I've only done one of my two so far. But was on the regulatory environment, so that was always fun of how you get drugs approved or even just in this case like stem cell products which are used in clinical trials. How you get those approved for GMP manufacturing and what that means, and a lot of myths. So myself and the guest went into the weeds a little bit and drug Nicole in there a little bit on that, because it's a very particular environment being in the regulated space and knowing that it's its own language. But it was very fun, and we nerded out, but that's to be expected.
I can imagine. Right up your alley, Jason. Well, I had really nice discussions. I already recorded both of my episodes. And really nice discussion about, global science. We had some where our guest comes from a big scientific kind of initiative in Africa. So really nice for people like me that don't come from Europe/America. And also, about getting funding, finding funding for your research on using tools on a global scale. What can you actually kind of funding you can find? Especially again, I think it's very interesting for people in maybe not in the highly developed countries, but I would hope that it will find it very educational and very useful. So so March 5th, you can find the new season of Lab Coats and Life. But well, until then. This is episode 99, by the way. How cool is that?
It's almost cool as episode 100.
I know. And I mean, can we say, so we are very excited about today's guest. We have a wonderful conversation up for you to enjoy with Luke O'Neill from Ireland, from the Trinity College in Dublin. But our guest for the 100th episode is, do you want to do? Should I do some drum roll? Nobel Prize winner Peter Doherty. Yeah, the guy with the institute named after him. How cool is that?
It is cool. It was a very fun conversation. And episode 100, we went a little extra long just for the audience. Milk the celebration, as it were. But the show must go on. We have papers. I'm going to do the one of these is easier to talk about the other. So I'm going to start with the hard one first and get it out of the way.
Okay, I'm ready.
All right. It is phages carry orphan antitoxin-like enzymes to neutralize the DAR TG1 toxin antitoxin defense system. It's in Nature Communications, coming out the 13th of February. The first author is Anna Johannesman, and the last author is Michele Leroux. And I will say I learned a lot reading this one. So, there's a constant war between bacteria and phages. And so apparently bacteria pump out toxins to attack phages. One of these is the DAR T1 toxin, which is an ADP ribosyl transferase. So it ribosylates the DNA of the phage and prevents it from thus being able to get replicated. So it just keeps modifying DNA, and then the phage can't do its thing. Okay. So then phages counter this with an antitoxin, which is a ADP ribosyl glycohydrolase. So it undoes the ribosylation. And so there's a war going on in the microbiological sphere between phages and bacteria with all of this. So we all we all love CRISPR today, but as a reminder, CRISPR is an antiviral system in bacteria that we adopted for, genetic engineering. But so the notion and understanding this has a lot of potent value. What they found here, and this is where I don't understand how big of a deal this is because I'm not a viral phage person to go, ooh, this is really neat. But it is really cool just generally, is they found that there's this orphan antitoxin toxin system going on, or really antitoxin in the T4 phages that they found some interesting results. So T-even phages, because apparently T phages 2, 4, 6 are all similar. I didn't know that. Do not rely on a previously known antitoxin for this DAR toxin, right? They don't have it. It don't work. If you get rid of it and delete it out, they still survive. So they have something else going on. So they then did a screen and figured out that there's this other gene that's really important, and identified eventually a new antitoxin gene. They did some CRISPR, screening, so to speak, and then using E. coli as well as the model organism for the bacteria. And they identified that this new thing, which is ADFNT4, which is a nadir type. Apparently, that's how they define these, is it's a nadir type antitoxin, is the primary antitoxin for this. And it is indeed a nadir family DNA ADP ribosyl glycohydrolase. So they do all the work to find this out, looking at plaque sizes, knocking things out, tracking where it is on genomes, crossing things so you can do, viral recombination. And then, when it recombines and now it's surviving or not surviving, they do those types of techniques you'd expect to be able to get down to, it's this bugaboo. All right, so they get the bugaboo. But here's what's wild. It's really only effective in this strain, in these T-evens. So it's specific for activity in the context of this tenavirae. So it can't stand in for DAR1 and E. coli. If you take it out of the virus system and just try to have it function elsewise, it doesn't work, which is not typical. So that's one really weird thing. So this one, the specific one they first found, usually you can pull it out and it still has its regular activity and it doesn't, and they don't know why. They couldn't figure it out. But then they found that other bacteria have a very similar gene, or not, sorry, other viruses that are evolutionary pretty close have similar genes, and those that are also active, ADFN BAS32 and BAS60, those can be pulled out and function just fine out of the T4. So what they're showing is that this seems to be that this protein itself with small evolutionary changes seems to have been spread, and it's important for resistance to phage toxin generally in this family of viruses. But the one they started with in T4 that they started the paper with, unlike its close brothers and sisters, doesn't operate outside of T4, and they don't know why. And that's where the paper ends. But it's interesting that like everything else behaves normally and it loses its function. And they think they can't figure out if there's like some other genetic component or cofactor or anything. So
What a mystery.
there you go.
I I'm a bit surprised. I want to sound very ignorant here, but I'm very surprised that there was a whole gene, there was a whole protein sequence, and they did not know what it was. But how is that? It's not such a large genome that a phage has, right?
No, they're not small, but they're not large nor small, right? It's not like a plasmid, right? I believe the phage genomes are bigger than itty bitty.
Yeah, but I find it kind of strange that by now we don't have like every single.
Phage sequenced.
Yeah.
There's too many. It's like we don't have every single bacteria sequenced.
Yeah, but T4 is like the phage where we get our ligase from, right?
Yeah.
So I would feel that I would have
Well,
thought that
they knew
that
there was a gene there. They just didn't know what it did.
yeah. Okay.
They had to bust out AlphaFold and then figure out that, oh, it looks like one of these other things, and went.
Ah, okay. So they have like the coding. What's word? I'm looking for a word. It's escaping my mind. I feel like a coding sequence. There's an origin of initiation of transcription, translation, and you're like, well, there's a gene. There's clearly some protein being made from here, but they didn't know what was the purpose of the protein. So that's what you're telling me.
Yeah, like they didn't know. Like, yeah, there's a protein here. Great, there's an open reading frame, and
Open reading frame. That's the word I was looking for. An open
a
reading frame.
hydrolase of some form, or an ATP box, or what ADP. But like, what does it do? Oh, it's the
Okay.
principal antitoxin,
Oh, that's very cool.
but it
All
doesn't
right.
work outside of its own organism, or on its own virus, unlike most other antitoxins. You can
Yeah.
transplant them, you can put them in E. coli and watch them have activity generally, and it does not work.
So it must be interacting with something else from the virus. Must need
Something,
something else.
but who knows what that is?
All right. Okay. Well, then we can move away from, evolutionary race between phages and bacteria and move up the evolutionary ladder to uh, gnosis. Yeah. Yeah. That makes sense. My paper I'm going to talk about first is called Granzyme K expressing CD8 T cells promote recurrent airway inflammatory diseases. So it's published in Nature on the 15th of January. And there has 4 first authors and 4 corresponding authors. All of them, or most of them, at Tsinghua University in China. And I'm going to try to pronounce their names without making a fool of myself. So we have first shared authors, Feng Lan, uh, Ji Shou Li, Wen Shan Miao, Fei Sun, and corresponding authors Xing Liu, Xuan Bin Huang, uh, Luo Yang, Hai Chi are the corresponding authors, as I mentioned, most of them in Tsinghua University in Beijing. So I think um, this is a very interesting paper in which they are looking at kind of patients that have recurrent nasal polyps due to chronic rhinosinusitis and asthma. And so there's inflammation in the upper airways. And there's some patients that just keep developing nasal polyps, and this, makes them harder. It makes it harder for them to breathe properly. So they get uh, uh, a surgical a resection, and some people get it again and they get surgically resected again. And so these researchers took advantage of samples coming from patients that had their nasal polyps removed, and a few patients that had it done twice, and they decided to look into the T cell repertoires of the T cells inside those tissues. So they isolated T cells, did some single cell, no, they did some bulk sequencing at the beginning, and they find that there's certain these are some single cell transcriptomic analyses, and they see that one of the first things they notice is that in sequential surgeries in the same patients, there are a few clones that are, very present both in the first and in the second resection, suggesting that there's kind of, population of T cells that are, resident there, and they just, keep colonizing this tissue throughout time. And they find a most of these clones that were repeated, they belong to some kind of effector memory T cell population, which was different from another population that was more like tissue resident. But in this case, they find a when they do kind of single cell analyses and they find different clusters, they focus on one cluster of cells that have one of their characteristics to express granzyme K. And granzyme K is a serine protease that is expressed mostly by CD8 T cells, also NK cells. And unlike its, well-known granzyme B cousin, which is, part of the cytotoxic granules of CD8 cells and induces apoptosis in target cells, granzyme K does not have cytotoxic functions. It actually is a non-cytotoxic granzyme, and it actually, cleaves proteins. So they find that patients with more severe nasal polyps or more comorbidity of asthma, they kind of tended to have more of these granzyme K positive CD8 cells. And though when they think about, well, what is this granzyme K doing? One of the things that when they sought to identify, molecular targets of granzyme K, there's one particular kind of set of proteins that this enzyme could cleave, which is components of the complement system, particularly I think C3 and which really activates the complement cascade, and this drives inflammation. And so they see that they show this that granzyme K can cleave the C3 and generate C3A, and this is really promotes immune cell recruitment in the airway. And what is also interesting is they show with immunohistochemistry that there's kind of this tertiary lymphoid structure that are formed in this nasal polyp tissues, and really kind of suggesting recruitment of different types of immune cells, and this sustains this chronic inflammation. And so they moved into a mouse model of asthma, and they sensitized mice with OVA LPS, yes. And then they have kind of repeated challenges with aerosolized OVA just to kind of induce this asthma syndrome. And they show that there's an increase in Granzyme K positive CD8 cells in the valve, in the bronchoalveolar lavage fluid, so like the fluid from the lungs. And as if they deleted Granzyme K and T cells, this actually really reduced airway inflammation, which included fewer recruitment of eosinophils, neutrophils, dendritic cells, and improve lung function. And they also did some adoptive transfer experiments, and they show that if they have CD8 T cells that come from Granzyme K knockout mice, this failed to although this really reduced the recruitment of eosinophiles and kind of inflammation in general. And conversely, overexpression of some Granzyme Ks that are overactive Granzyme Ks, but not, for example, Granzyme B, increased airway inflammation, kind of really showing that, Granzyme K expression does seem to be a causal for this inflammation. And so basically the idea that they suggest is that maybe this means that you can target Granzyme K in asthma or in like inflammatory upper airway disease. And so they do a different system in which they can delete Granzyme K expression with tamoxifen. So they have this, this CRE-ERT2 mice in which if you cross with a CRE flux mouse for Granzyme B, then you can induce it with tamoxifen. And then they show that even after the disease onset, they could reduce airway inflammation through deleting Granzyme K. And they have there's one particular inhibitor that they look into, PPACK, and that is a Granzyme K inhibitor. And this seems to reduce airway eosinophilia and tissue damage. Whereas, inhibiting Granzyme B, again, did not do the thing. So I think it was really cool because I don't think you think Granzyme K is what is Granzyme K doing? And, this ability to cleave complement proteins really seems to mediate a lot of how T cells can initiate this response and, really support inflammation. And I think it's really cool because kind of started with, material from tissues from patients with a very specific disease. And so it's a really cool paper that I read and I shared with you today, Jason. I hope you liked it.
I do. I think, airway disease is a big problem. Granzyme, I like Granzyme because it's druggable potentially.
Right. Yeah, that's very, I think that's really nice when you find something that you can actually do something about.
It's I found this additional transcription factor with no known inhibitors that is important for my disease. They're excellent, but
good for you. Yeah. So I thought that was pretty cool. I thought it was pretty cool.
Yeah, I like it. All right. It's been several weeks since I've done something in the intestine, so we're back.
Right back at it, baby. Okay, what you got?
Science Translational Medicine. Intestinal epithelium-derived IL-34 reprograms macrophages to mitigate gastrointestinal tract graft versus host disease. It came out 12th of February. All right, first authors, Aditya Rayazam and Allison Moe, and last author is William Drobsky. So, graft versus host disease, right? You transplant your bone marrow because you have cancer usually, get rid of the old bone marrow, put some new bone marrow in, and then your immune system decides to attack you, and intestinal or gut or GI acute graft versus host disease is responsible for the overwhelming majority of graft versus host disease, with its pretty significant mortality. If you get it, you have a 50% chance of survival or something, depending. It's not good. So figuring out how to stop that would be really great, right? So this is mostly in mice. They did a little human work at the end. They noticed the first thing is that macrophages in the GI tract during acute graft versus host disease. And by the way, this is just a very standard model. You do a bone marrow ablation of one mouse and put a mouse that's not syngenic in, and it gets graft versus host disease, right? So you can give mice graft-versus-host disease pretty easily, which is nice, right? It's actually a relatively practical animal model, all things being considered. So there's transcriptional heterogeneity. Some are pro-inflammatory, some are anti-inflammatory. Please read if you want to look at all their single-cell RNA-seq, which is, par for the course these days. So they notice that there's a increased percentage of these in mice with graft-versus-host disease of the receptor for IL-34, which is CSF1R. You're like, does that mean IL-34 is important? So if you take IL-34 donor cells and put them in a regular mouse, they don't have any difference. If you take regular cells and put it in IL-34 knockout mouse, the mice die more. They have worse graft-versus-host disease. So this is the nidus of the paper, is that recipient mice without IL-34 have more inflammatory macrophages and less suppressive macrophages, and that there is a strong signal there that they then start exploring. So they do some more single-cell RNA-seq, and they identify this to an extent, and I'm going to spare that as well. But the important thing that they find is that the IL-34 in the recipient regulates the accumulation of CD4 positive GM-CSF positive T cells in the colon. It tamps it down. It holds it in check. They also then do studies, what compartment matters here, right? And they establish that it's the intestine with tissue-specific knockouts and a BGAL reporter system. They establish that it's IL-34 in the epithelium of the recipient that's really important. They also do kinetic studies, and here's what's really important. It's the conditioning that primes everything. So before you do a transplant, you irradiate, you do chemical conditioning, what have you. That is when IL-34 is turned on in the damaged intestine. It tamps down the intestinal damage and suppresses the intestinal immune system with more, suppressive macrophages. And having a good response there by the IL-34 leads to less graft versus host when the new immune system's introduced, because it causes less homing of pro-inflammatory T cells into the intestine. So that's the story. They show that recombinant IL-34 is protective, that it's the IECs producing it, and that in the donor macrophages, the IL-34 it involves ApoE. So ApoE on the macrophages is somehow, and they don't get all the way down, but somehow involved in this. They see it on the single-cell RNA-seq signal. If they knock it out, IL-34 does Bo Diddley anymore. It's not protective. And so ApoE is a critical protein in the macrophages that responds to the IL-34 signaling. And then they see that, in humans, it's not exactly the same setup, but they see that there's this IL-34 in epithelial cells that are adjacent to macrophages, and that ApoE is also upregulated in these macrophages in graft versus host intestines samples from humans. And then they're done.
I mean, that's really cool because, of course, the intestine is a major target for graft versus host responses. And I guess, as you said, because of the conditioning, there's a lot of damage done to those very fragile, highly proliferative cells. And that's usually where a lot of these, if you can stop the graft versus host disease at the gut, then you can really prevent it from spreading to other organs or from getting worse. So that's really important. And so the macrophages, how they is there a phenotype that associates with them, being with this IL-34 presence? So when it comes to I think you said anti-inflammatory, but what exactly is it that they're making?
So the intestinal epithelium is making the IL-34, which signals through the receptor, that the CSF-G1 receptor on the macrophage, but ApoE is involved in the effect to tell it to cool the hell down and stop. Those macrophages signal and become more suppressive, and then do not pull in all the T cells.
All right. Okay. So I say maybe you would expect like less infiltration of T cells
You
in
see less
the.
infiltration of T cells.
Okay. All right. That's very cool. Well, I hope that they can do something with that. Again, in the clinic maybe. All right. So, talking about macrophage, well, that's our a common theme for these two papers: protective macrophages. I think that throughout time, macrophages have always gotten more of a bad rep. They're this garbage bags eating stuff and activating cells. But I think that protective macrophages are all the rage this lately. So I have another one. You tell me, protect against GVHD, and I double down with macrophages protect against sensory axon loss in peripheral neuropathy. That's the title of the paper published in Nature, 12th of February. First author Sarah Hakim from the lab of Clifford Wolf at Harvard Medical School. And um, in this paper, they are looking into a complication from type 2 diabetes, so the metabolic diabetes, so to say, that is pretty prevalent, affecting about 50% of patients, which is peripheral neuropathy. And this leads mostly to sensory loss and neuropathic pain on limbs, I guess, of patients with type 2 diabetes. And it's not clear what is at the immunological level, what's happening here. And they want to study the contribution of particularly macrophages on the onset or progression of diabetic peripheral neuropathy. I have to say that I really like this paper, but I don't think they end up really explaining much. They do highlight a very interesting population of CCR2 positive macrophages that seem to be protective of against axonal damage. But they don't really go into like I think the origin of the neuropathy is not completely clear. And it's yeah, so all they show that this but basically they show that the persons of the macrophages protects against progression. So let's talk about the peripheral neuropathy. So basically, um, they have mouse models in which they have high fat, high fructose diet, I think it is. Um, yes, and basically they get obese mice. And these mice seem to mimic what they observe in human patients. They develop sensory deficits. And some of the measures that they do is what is known as hypoalgesia, and that means the loss of they're unable of like the loss of sensitivity to heat. Uh, what is called as mechanical allodynia, in which they feel pain from non-painful kind of mechanical stimuli, and and this is kind of what they use to measure axonal, like the damage in the axons and in this sensory neurons, and they show that they can give this through this high fat, high fructose diet to the mice. And um, how is it exactly that this comes to be? It's not. I don't think it's completely clear to me. But basically, the metabolic changes that are brought in by this high fat, high fructose diet seem to disrupt neural function through accumulation of toxic lipid metabolites in nerves. So this is what they call dyslipidemia. Also, certain insulin resistance that is also alters the metabolism of neurons, problems with oxidative stress, and this all kind of negatively impacts the peripheral neurons. And also there's one thing which is a gene called SARM1, which is a regulator of axonal degeneration, and this gets kind of this gene seems to be increased in response to metabolic stress. So these are kind of the ways by which this high fat diet basically affects neurons. So that with that, and this is something that they can see in mice. So they what they do is they look into the progression of the disease, and I think what's important is that they see this allodynia, this hypersensitivity, kind of peaks at 8 weeks, and then it develops into hypoalgesia when they stop feeling. So it's like you have a peak of like pain, and then at 12 weeks, this is gone, and uh, they have other kind of measures of axonal degradation, and they kind of also kind of progress through the different weeks. When it comes to the immune cells, they find that when they do single cell sequencing, and they show that in the sciatic nerve of these mice, which is kind of like where these neural axons come from, there's an increase in CCR2 positive macrophages at 2 weeks, which is before they see significant loss of the axons in these mice. And these recruited macrophages, they look at the transcriptome, and they seem to have genes associated with nerve repair, with neurodegenerations. This is similar to macrophages that appear after a traumatic nerve injury. So there's this crush test that they do. They can crush the neurons, and this, initiates a repair program. And the deletion of CCR2 or the pharmacological inhibition, which depletes or the recruitment of macrophages, worsens the progression of the hypoalgesia and the skin, and the degradation of these neurons on the skin at 12 weeks, for example. And a very important gene, galectin-3, if they also knock out that gene, if you loss of that gene, also increases the progression of neuropathy. And this is galectin-3 is a gene that is expressed on the CCR2 positive mice. So basically, they suggest that this macrophages that are found in this high fat diet of mice, they are actually protective of the neurons, because they're also observing other situations which you need to protect after injury, and the removal of these macrophages increases the speed of degradation of damage of the neurons. So what they say is that they suggest that this galectin-3 helps through removing debris. If I remember correctly, so removing debris from the damaged neurons, and this kind of helps them increase their viability. And so that seems to protect the neurons from the high fat diet and the degradation they've seen in this model of type 2 diabetes.
It's interesting that they can get peripheral neuropathy to occur in the mice. I wasn't sure how human that was, if that makes sense.
Mm.
And I wonder what the macrophages are doing.
Yeah, so basically they suggest that their neuroprotection is this is quote from the paper, by virtue of clearing debris, excess fatty acid, or metabolites such as deoxysphingolipids, which accumulate in nerves.
But they don't know.
No, they do show that Mike lacking this GALF3 exhibit more severe symptoms. So particularly the heat, they focus they rely a lot on the heat hypoalgesia, which is this loss of sensitivity to heat. And because they can this GALF3 recognizes several products that are associated with damage, so they can really take care of this debris and remove it so it doesn't generate more damage, if that makes sense.
It does. So we're going to talk about macrophages more
Yeah, that's true.
with Dr. Luke O'Neill at the Trinity College of Dublin in just a moment. But before we get to that, we're not at one place this year, but two, because we're going to be at the IUIS 2025 conference that's coming up this summer in August in
Whoop,
Vienna.
whoop.
Our last episode, number 98, goes into plenty of detail about what to expect at the conference and features keynote speakers. So definitely take a listen to that, and hopefully we're all going to see you there. You can find the program at iuis2025.org.
For the second part of our show today, we're very happy to welcome Dr. Luke O'Neill. He is professor and chair of biochemistry at Trinity College in Dublin. And the O'Neill lab has really interesting work on molecular basis of inflammation. And I would say personally, from my own scientific experience, I'm very much looking forward to talking about the contributions that Professor O'Neill's lab has done to the field of immunometabolism and understanding how metabolic routes affect particularly macrophages. On top of that, also our guest today is a fellow podcaster. Professor O'Neill hosts a weekly show called Show Me the Science on the Irish National Radio, and also author of books. So I think we're going to have a beautiful conversation today. Luke O'Neill, thank you so much for joining the Immunology Podcast today.
Oh, thanks for inviting me, Brenda. I suspect we will have a good conversation given our many common interests that we share already. So.
Thank you for being here. All right, Brenda, go first. It's immunometabolism.
If I may start with that, because I think I witnessed a lot of the, big discoveries in immunometabolism last year. So was very looking forward to this conversation. Let's talk a bit about a lot of your work has been done on macrophages and understanding the role of different metabolic routes in macrophages, not only for macrophages to get like energy, but also like all these secondary metabolites or all these metabolites that are part of, for example, the Krebs cycle, glycolysis. They actually have all these kind of tentacles of influence. And I think it's very interesting to put that in context. So maybe let's start about, what immune cells take as kind of substrates. It's not only for energy. If you think glucose, glutamine, other kinds of substrate, it's not only about energy. There's so much more. So what would be your highlights to understand the importance of metabolism in programming immune cells in general?
Well, it's a surprise, Brendan, this field has emerged in some ways. When I began in immunology 35 years ago, there was no talk of metabolism really in the immune system. It's amazing, And the main reason for that was immunologists didn't like it, because it was like, Krebs cycle, what the hell is that? That's biochemistry, So it's a funny one. And then it began to emerge. I think it was Diane Mathis actually coined the term immunometabolism. And what she was working on was more things like obesity or diabetes and how the immune system talks to those sorts of metabolic situations. And we call it kind of systemic immunometabolism. And it is very interesting. I think in obesity in particular, you see loads of gamma delta T cells. My great former colleague, Lydia Lynch, is a real pioneer in that area, So that was interesting anyway. But my side was a bit different. It was intracellular. So it was metabolic change during immune cell activation is what we've worked on for the past, I'd say 12 years now. My lab kind of shifted focus away from signaling, because I was a big signaler for a long time, into metabolic change. As a signal, though, I think the important, as you said, is it's not just about the boring aspects of metabolism that we all learned in biochemistry. These metabolites are doing very interesting things, and they're talking to transcription factors. They're affecting the epigenetic landscape, And then lo and behold, in the case of my favorite one, itaconate, for instance, it's modifying proteins that are key for macrophage function, So suddenly all this vista began to emerge. I think one big reason for this was high resolution mass spec was invented. You could see the damn things. And because you got to see them to be able to study them, don't you? And then we began to see metabolic sort of specificity in terms of some of these changes. So I think what's happened in a nutshell is we've made metabolism great again. How about that?
No.
but certainly, for me personally, and many of us in the field, yourselves included, there's a whole treasure trove of new things to be found out here in immune cells, And I guess if you're an immunologist, what it really means is the metabolic landscape governs the phenotype of that immune cell. And that was unexpected. Like we thought Krebs cycles everywhere, boring, forget it. not the case. You will see very specific changes in these pathways depending on the cell type you're interested in. And obviously, as we discussed, Brendan, from your work, I guess, with Tim and so on, T cells, NK cells, it's not just macrophages, they all are showing metabolic changes that are somewhat specific, shall we say. And I think overall, we're at the beginning of this. Horrifying as it may seem complicated now, but come back to us in 10 years' time, we'll have even more complexity to contend with, I think.
So I had a question here on this end. So we know like classical signaling, right? There's a cell surface receptor, and let's just pick NF-kappa B, right? And it goes in, and you're going to have TLR signaling, and then downstream NF-kappa B is activated, which leads to transcription. So is the immunometabolism, these metabolite pathways, a parallel pathway, or have you found that the signaling converges largely on metabolites? So is it, I guess what I'm saying, is it another set of signaling just like all these other sets? Or is there some master component here because it is the metabolism of the cell and it's really in charge?
I wish it was just signaling, Jason, that'd make it easy, wouldn't it? I mean, because I originally worked on TLR signaling, for instance, and we discovered MAL, one of the adapters. And it turned out that's quite simple, because here's a receptor. You add LPS, as we know, to that specific example. You bring in MAL and MyD88. You eventually end up with the IKKs. You get to NF-kappa B. It seemed complicated at the time, but now it's just a linear pathway culminating in NF-kappa B. Now, of course, NF-kappa B then will regulate, I don't know how many genes it is now, but it's probably thousands of genes get turned on by the NF-kappa B system. So it gets complicated downstream because all these different genes get regulated. Now, metabolism is coming off these pathways. I mean, that's how we got into it. If you add LPS to a macrophage, it makes a huge amount of lactate. It makes so much lactate, the medium goes yellow. And in my early talks, I used to show a slide of a control macrophage in a Petri dish. The medium was nice and pink, LPS has gone yellow. So something that got my imagination. What's going on there? It is another LPS response, just like NF-kappa B, but yet it's a metabolic response. And then the question became, can we map that? Can we figure out what the metabolites are? And then once you see these metabolic changes, what are those metabolites doing? And they're doing so many things. That's the challenge, really. So it's coming off the signaling pathways. I mean, you need NF-kappa B, you need HIF to drive expression of some of these enzymes in glycolysis, for instance. So it comes off the signaling pathway, but then it gets complicated very quickly with all these multiple metabolites changing. You can end up with a simple enough scenario sometimes, though. So one of our early papers saw succinate go up, right? And then we link succinate to HIF, and we could show you needed succinate to drive HIF as a transcription factor, and then you get the output from that. And that makes succinate a signal as much as anything else, because the succinate has to change. Signalers are very binary in their minds. It's the control cells and the activated cell. What's the difference? And the difference is all these components being turned on, and metabolites are another difference between those two states. So it's probably overly simplistic to call them signals, I think, because there's a lot more going on here. But effectively, it gives rise to an output that's no different to a classical signaling pathway.
It's really cool to see how we came from, to say, the Warburg effect. And it's just consuming glucose, and then you have all these acidification in the media, and that's it, to actually showing that so many of these intermediate metabolites act as signaling, but also act to support the metabolic requirements. It's not necessarily fuel, not necessarily to get ATP, but to get building blocks, to get the right amount of protein synthesis. So when you talk, for example, in the case of macrophages, which is your cell of most interest, how do we understand LPS activation in terms of metabolic reprogramming? So what is the current kind of understanding around the effect of metabolic reprogramming for, let's say, inflammatory macrophages?
Yeah, so we know a lot about that now, as you know, by now, over the last 10 years or so. Many papers have come out on this. So if you use LPS, the classic trigger, any inflammatory trigger really will do this. Glycolysis goes up. Let's start with that. And you can see all the enzymes get induced, and GLUT1 gets induced, and hexokinase. So you ramp up glycolysis. That will have two effects. You will make ATP from glycolysis, as you may remember. Not very much, but you make a bit of ATP. And the fact you can ramp it up so quickly means it becomes a major source of ATP. So even though in your biochemistry years ago, you would have learned how Krebs is the TCA cycle makes loads of ATP per glucose molecule, like 36 or something. Glycolysis only makes 2 net. You would think Krebs should dominate, but it doesn't in a macrophage, strangely. Glycolysis does the heavy lifting. And the reason for that is to make more of the TCA cycle, you've got to make more mitochondria. And that takes a long time. That takes too long. So the way we think of it is in an acute injury, acute inflammatory situation, that macrophage needs ATP to fulfill its bioenergetic roles and fuel the whole system. So you ramp up glycolysis to achieve the ATP production. That was the first thing that was really seen. And that had been seen back in the 70s. People were able to measure glycolysis in those days. And there was a guy called Eric Newsom in Oxford, and he was using macrophages. And Simon Gordon, actually, who was the macrophage pioneer, they published a couple of biochem journal papers, but they thought it was just ATP production. That was their main conclusion. Now, secondly, you get off glycolysis, you go into the pentose phosphate pathway. That will make, for example, nucleotides. And you need the nucleotides to replicate the DNA, and the macrophage can proliferate to some extent, You also make NADPH, very interestingly. That's for the oxidase to make reactive oxygen. So again, that's an obvious macrophage type response. And then other intermediates come off the pentose phosphate pathway for different metabolic processes as well. Now, when you get to the TCA cycle, as the Americans call it, that's a bit different because that gets very complicated very quickly. Early on, you boost OXPHOS, actually. You can increase flux through the TCA cycle to make ATP. But over time, that gets remodeled. There's 2 breaks overall. There's one at succinate, and that's very clearly been seen now. But we were the first to show that, but other labs have similarly shown that succinate now builds up. Now, succinate will have 2 effects. It activates HIF, but before it starts to unravel the Krebs cycle, it does get oxidized through succinate dehydrogenase, and then you get what's called reverse electron transport from complex 1. Now, that's really weird, and we published that in 2016. So the macrophage, in its wisdom, runs the electron transport chain backwards, right? Now, when that happens, complex 1 makes ROS. So the actual output from that is reactive oxygen. Right. Now, that's been known as a long time signal. We've known about ROS going up and all these different things. And ROS will do many things. It does activate HIF. Another way to activate HIF is through that ROS production, right? Now, the second major change you see, and I think this is our most, I hate to say the word important because it makes me sound like someone who's important. But I think it's the most profound thing as our taconate goes up. And that comes off aconitase. Now, aconitase is normally in Krebs cycle turning around. We get a diversion. And this, in fact, is the best example of metabolic reprogramming. So in the resting macrophage, you can't make a taconate. The enzyme that makes it is not there, or it's too low. It's called aconitase decarboxylase. LPS induces that. So do type 1 interferons, actually. They're great inducers of this enzyme. And now you rip aconitate away from the TCA cycle, you start making itaconate, and that goes through the roof. And in the murine system, that gets to millimolar concentrations. It's a huge diversion. The question then becomes, what is itaconate doing? And in the last year, it's the gift that keeps on giving. Let's put it that way, because all these papers have come out. We published our paper in 2018 on this. It's nearly 7 years ago. And it was, at that time, there was only 4 or 5 papers on it. Now there's hundreds and hundreds of papers on itaconate. And it does many different things, and hence the complexity, I suppose. It will block SDH, so it blocks that flux from succinate, and that will limit inflammation because you lower ROS in that situation. That's one thing it does for definite, but equally, it does modify. Many proteins on cysteine get modified. It's a cysteine reactive molecule. IRAK4 is our latest paper, actually. This is a big collaboration with Eli Lilly. I'm very happy to mention because they're funding a postdoc in my lab. We had a paper with those guys, Luke Borner, Chuck Dorsey. They did fantastic proteomics, and they found IRAK4 was being modified on a key cysteine, and it turns IRAK4 off. Now, we know from our previous discussion there with Jason, IRAK4 is a key signal for TLRs and IL-1 and IL-18. Here's a natural inhibitor of IRAK4, and that was shut down inflammation. And that's one example. NLRP3, one of my favorites. JAK1, that kinase in the famous JAK-STAT pathway, that gets blocked by itaconate as well. And so suddenly you see this negative feedback effect going on with a macrophage coming off the TCA cycle, most unexpected. In fact, when we saw itaconate go up, we didn't know what it did. We just observed it rising, you know. It could have done anything, you know.
Yeah.
We linked it to NRF2 in the end, actually, because it can drive NRF2. Now, there's a bit of controversy in this field, though, because there are derivatives of itaconate that are more potent than the endogenous molecule. And we showed that with one we made called 4-octyl itaconate, and that's true. So the endogenous molecule probably does less than the natural, or than the derivatives. But most importantly for us, we've made a drug out of it. And with Eli Lilly, we have a drug now in the clinic based on itaconate that's profoundly anti-inflammatory. You know? So in other words, we made a derivative, we end up with a potential new anti-inflammatory molecule. And now Lilly have just finished a phase 1 trial. They've announced they're going to start a trial in an unspecified autoimmune disease, which we can't disclose. But they're going after it, you know, as a therapeutic. So I'm delighted by this thing. And imagine what a thrill this is. I don't care if the natural molecule differs. We got a drug out of it. You know?
So they haven't formally publicly announced the condition yet for it?
No, you see it on the website, Jason. It's an unspecified autoimmune disease.
Yeah, that means they haven't registered with clinicaltrials.gov yet.
You don't need to be a genius though to figure out which one it is. I'll give you a big clue. So dimethyl fumarate is a drug that's anti-inflammatory. It's a fumarate derivative.
Yep.
That was discovered in the 70s, if you can believe it, as a way to boost the TCA cycle, almost like just give it a go.
Yep.
DMF is called Tecfidera. It's approved for psoriasis and MS.
Yeah.
It's a multi-billion-dollar drug for Biogen. That was one of their lead products, you know. Yeah. Now, we've made the case, and the evidence would support this. DMF is mimicking itaconate. You know, in other words, itaconate's cysteine reactive, so is DMF. So we think not just us, I mean, some of DMF's clinical efficacy, as it were, or the basis for its effect is probably cysteine reactivity, just like itaconate, you see. So, yeah. So we're in the world of DMF here, is what you would say.
Being in pharma myself, I also wonder if they don't know yet. Like often these anti-inflammatory drugs,
yeah,
after you do your phase 1, you're going to go and say, well, there's 15 conditions I want to go after eventually. Which is the one that's going to get me to market first? And then you roll out. So you'll see like all these drugs, they'll go after either MS or psoriasis or IBD.
You've got it.
And then they go after the other ones after they do the first one.
It's not very scientific at one level, you know.
But the science is which do you do first because of the right indication and the market and the trial design and what will the FDA require for that very first trial. And then it's much easier once you're on the market to expand out to other indications. And so there's the first shot on goal really matters. And so I bet you that's where they're at. You've got it.
And remember, many trials fail for the wrong reason, you know. So it can be PK, PD, all these issues come up. And I mean, it's interesting. But you're right, though, Jason. I mean, what I would say more generally, all these great discoveries in the past 10, 15 years in immunology, right? Not just in this business that I'm in, and in many aspects. We're getting closer to the panacea. I know it's a broad claim. Like and my work on the NLRP3 inflammasome, for instance, that there are 27 NLRP3 inhibitors out there now. 9 are in the clinic, and you could list 20, 30 indications where an NLRP3 inhibitor could work, you know. And of course, we know there can be a panacea because of steroids. Steroids actually are a panacea for inflammation, but the trouble is they got awful side effects. So in fact, on that, a very interesting paper last year by Gerhard Kronke's lab, it was in Nature. Guess what steroids do? They increase itaconate. So there's some evidence now that, and steroids don't work especially well in the IRG1 knockout mouse, which can't make itaconate. So we know itaconate's tied into some of steroids effects in a way.
So I've learned more recently that, you know, we always think steroids are all through transcription, but that was the old view.
Yeah.
Now there's an understanding that steroids also have kind of an acute phase anti-inflammatory, which clinically we knew, like people felt better more than 24 hours later, which is the transcription part, but they really feel better 24 hours later. So there was like the main mechanism very clearly delayed transcription, but something was happening earlier. Is itaconate the earlier part?
Could be, John. In fact, what that paper shows is amazingly, the steroid receptor is binding pyruvate dehydrogenase. There's a great enzyme for you, right?
Oh, I love it.
That's the bit that's surprising. You think they would have discovered that 50 odd years ago. So it's binding PDH, right? When you add the steroid, the receptor dissociates, goes to the nucleus to do the transcriptional part, right? Freeing up PDH to increase flux into itaconate. That's what the paper shows.
So maybe that's the acute phase. Maybe that's the 2 to 4 hour. Because there's definitely, they've now acknowledged a 2 to 4 hour effect.
Yeah, you got it. You've got it. And of course, the dream there would be can you replace steroids with an itaconate derivative that wouldn't have the side effect? That's the direction that's heading in. Now, there's been many attempts to do that have failed, of course. So I wouldn't overreach here, but it's certainly a prospect that's worth testing in the clinic. Let's put it that way. So we'll see how Lilly get on with their itaconate derivative in that regard.
Interesting. All right. So itaconate seems to be this master switch. When you talk ROS, I get it, right? Macrophages play with ROS, right? That's kind of their weapon in reality. So tying signaling back to ROS, I'm thinking about this from an evolutionary perspective. Makes tons of sense, right? Like, hey, I'm going to have some positive and NEDVAC feedbacks from ROS levels because that's my job and that's my weapon. So I want to understand how much of that I'm pumping out and have feedback to it. But itaconate, where do you think the evolutionary pressure for a lot of this immunometabolism that's clearly developed, but like, what the heck? Is this because in the end cells are metabolic machines and so it's the principal driver and it's almost like, not, I don't want to say vestigial, but early proto signaling because that's something that was always really there and all these receptors came later? Or like why? Essentially,
God, that's a high level question, Jay.
Yeah, I know.
I hear you. I mean, I think absolutely metabolism must have been the first signal because in the primordial soup, what are we going to call it, organisms, the single cell world, they took up nutrients and things had to happen, in response to those nutrients, and the cell might divide. Now, once cells become multicellular, then you might see metabolism changing with quorum sensing and release of metabolites to communicate. I bet that they may have well been the first signals for that, And then I think with itaconate, it is made in the mitochondria. There is evidence that itaconate can be antibacterial. You see, in fact, that was some of the early stuff on itaconate. It can kill TB. It inhibits an enzyme called isocitrate lyase in bacteria. So it wouldn't surprise me as if it was the endosymbiont it was the first thing to make it. And then it would get released from the endosymbiont and began to attack other bacteria, perhaps in again in a quorum sensing way, or possibly in a way to kill another bacteria that was a threat to the endosymbiont. And then lo and behold, the mitochondria go in 2 billion years ago, and they keep making it. And then it begins to adapt to other situations, maybe. So I think that's the way I think of it evolutionarily, perhaps.
And I guess kind of continuing with that idea, what are the risks of, if you have this kind of negative feedback at some point, what are the risks of not having it? Why would we want our macrophages to stop or to kind of tone down at some point? What is behind that?
Yeah, well, I see itaconate as the IL-10 really, in a sense. And we've known for a long time, LPS, somewhat paradoxically, is a great IL-10 inducer, remember. So even though it's a massively inflammatory molecule, it can drive IL-10. So and the IL-10 will feed back then and limit it. And that's well known. I mean, our latest stuff, actually, and you probably know, if you block IL-10, TNF goes through the roof from a macrophage. So it's a temperer of the word. Now, in this case, it's a metabolite that's acting in a negative feedback way. So it's another negative feedback loop. There's so many of them. I mean, clearly, if this goes out of control, you're going to get autoimmune disease or autoinflammatory disease. And that's well known, as you know. So in other words, I think there's many off switches operating here. And if the output's going to be a combination of the two, really, you see. So I think it's just another example of an off switch, really. The fact that it can target SDH is interesting because succinate is pro-inflammatory. Itaconate can limit that, and that kind of makes sense in a way because you have two metabolites that are juxtaposing each other. So I think that might be what's going on here. And as we all know, macrophages eventually resolve inflammation and cause repair to happen. And these diseases that we're talking about, they're defects in the repair process. So there's no doubt that these negative... That's why itaconate has a prospect as a therapeutic, to be honest, because it's mimicking a natural process, if you see what I mean. There's also good studies coming out showing itaconate deficiency correlating with disease severity. So, for example, in rheumatoid arthritis, there was a paper on this, low itaconate severe disease. So you're losing that negative loop in that situation and then somehow promoting disease. The trouble is, the caveat is we're oversimplifying it massively. Let's say that. You can again, the word overreach, you got to be careful. But these sorts of things are supporting this notion of a negative feedback loop, and in this case, coming off a metabolite.
I think that it's so important to talk about because autoimmunity in the end is probably evolutionary, the highest kind of source of pressure. So there must be, there's so many in terms of the immune system. Every cell clearly has some kind of inbuilt mechanism of negative feedback to prevent, because in the end, unregulated immune responses are so deadly, are so dangerous
Yeah.
for survival. So it only makes sense that we have several lines of defense against excessive inflammation.
Yeah.
And so that brings me to when it comes to other cells, we talk a lot about macrophages, no?
Yep.
But maybe if you want to
The
think
only
about
cell that counts.
the only
Not a-
cell that can, well, hey, don't leave T cells out of this. But are there other immune kind of interactions? Maybe it could be macrophages with other immune cells that are also kind of you find exciting or are particularly, we have learned something particularly unexpected.
Well, look at the beauty of the checkpoint with PD-L1, for example. And I mean, that's a great example. With the dendritic cells and macrophages, that goes through the roof. It'll turn off the T cell response. So we know there's always crosstalk between the antigen presenting shells, so we call it, and the T cell. And the T cell itself, as you know, is full of negative regulators, LAG3, TIM3, all those ones, So clearly, I think when we evolved adaptive immunity, that was a dangerous thing to do, because you're recognizing peptides that could be part of self. That was always the big thing, as you know. So we had to evolve ways to control this. Otherwise, so it's the price you pay for the adaptive immune response is autoimmunity, basically, And in certain people, it could be genetic, as we know, largely. It can be environmental, the combination of the two. Your adaptive immune system goes out of control and starts attacking your own tissues. Now, in the autoinflammatory side, of course, it's less adaptive, but still, if the macrophage goes out of control in that situation, again, you get an inflammatory consequence. So I think it is, as I say, it's the price you pay for having a very elaborate immune system. I'm pretty sure plants don't get autoimmunity, because if you don't have any adaptive immunity, you don't get autoimmunity, obviously. Flies don't develop arthritis, rheumatoid arthritis, so it's the price you pay.
What about the fact that there's two, like, I always think there's pro-inflammatory macrophages and anti-inflammatory macrophages.
M1, M2, oh no.
Oh, yeah, right.
Well, I now describe everything as a skid mark, so let's be very clear here. Like, there is, everything's just on a spectrum here. There's no binary, right?
Yeah, that's right.
But
very much so.
Thinking about the metabolism again, I know there's a lot of linkage between M1, M2, and metabolites and all of that, but it seems like in that case, then the outside world is pushing these macrophages potentially in unintended ways, right?
Yeah.
Like if I have too much glucose or I have too much downstream succinate, I'm going to accidentally pro-inflame all my macrophages, which makes me wonder, like, how we ended up evolving that part of the whole thing, which seems like a maladaptive evolutionary response, because it seems to just go wrong.
You've got to start with nothing is pathogenic here to begin with. In other words, as we know, the inflammatory process is meant to be beneficial. And it's the same with the metabolism. I mean, these things are changing in a normal inflammatory response, remember. When does it pivot into disease is the question you're asking, Jason, in a sense. Now that's a complicated thing to try to answer. I do think it could be nutrition. The big risk factors for these diseases are the usual things like obesity, lack of exercise, etc. And there's no doubt we're in the middle of an epidemic of autoimmune disease. And in fact, the incidence is going up, and it isn't just diagnosis, that's clear. It's too fast to be genetic, so it's got to be environmental. If you overload the system with glucose, you'll push some of these metabolites harder, and then you might see an inflammatory consequence to that. I think the other way to think about the M1, M2 business, those M2s, I mean, they evolved for two reasons. One was to repair and be anti-inflammatory, but to be antiparasitic. IL-4, IL-13, all the usual suspects are in there. Those kinds of pathways are more OX-phos, the mitochondria are respiring normally in response to a parasite. Now, why is that? Because you don't need a rapid response to a parasite. It's a more prolonged response. You can be more leisurely, So the inflammatory side of this is only important for acute infection and injury, really. So the cell, when you see the inflammatory macrophage, it's glycolysis banging away. In an antiparasitic response, you don't need as much glycolysis necessarily, and OX-phos suffices, you see. So again, I think that's another way to think of it. I think I always think of it through the lens of the host defense response, really, is what we're talking about, isn't it? and how that differs. But these are good questions, Jason, that haven't been resolved. I mean, it's a really interesting sort of a perspective to say, how did that end up in the way it did? It's not a bad idea to begin with infection and what the pathogens are and how they affect things, you know?
Yeah, like, I think pathogens, how is this the normal response? How is it becoming maladaptive now?
You got it. Yeah.
Is it maladaptive because we've changed the environment from where we've evolved?
Absolutely.
Or is it, or is there new pathogens now? Which I know some we think of this autoimmunity is because there's now pathogen mimicry of host, which and they're new enough that we haven't evolved out of that yet.
But the trouble is, there is a genetic aspect clearly to this as well. And as you well know, there's famous genes in the mix here that if you get them, you get a massive autoimmune disease, the famous Aicardi-Goutieres syndrome, in the autoinflammatory NLRP3. There's about, I'm going to pick a number, 15 genes, and if you mutate them, you get horrible systemic chronic inflammation. They're extremely rare, of course. Muckle-Wells syndrome is the one that we worked on. That's the NLRP3 one, but clearly you can get that as well. Now, what that might mean then for your average person, they'll still have variants in some of these things that might push them over the edge. So the maladaptive response will be partly genetic and partly environmental, inevitably. And our big hypothesis that we published last year, actually, me and a colleague of mine, or a collaborator called Mike Murphy, is it all about a break in endosymbiosis, really? So those environmental triggers are damaging the mitochondria, and they now reveal themselves to be bacteria, really, the enemy within. And lo and behold, it's no surprise then things like TLR9 is sensing mitochondrial DNA that gets released from the mitochondria, MDA5 in Rig I, that's sensing the double-stranded RNA coming out of a mitochondria. For that to happen, the mitochondria have to go rogue, you know what I mean? And it's the environmental influences that are making the mitochondria go rogue. And I've got a theory as well on this, that maybe those receptors evolved to sense mitochondrial nucleic acids, actually. And then they began to sense microbial nucleic acids. So there could be in that direction. You never know.
Clearly, there's a change in the environment, and again, because we did not evolve to survive the kind of diet or the kind of lifestyle that we have now, so there was no pressure to keep those those rogue pathways from developing as much as we do now. And
I think there'll be three things going on here, Brenda. Here's my grand hypothesis for today, especially for you guys. So number one, the genetics in this for definite, right? Number two, there's a lifestyle thing. We're no longer on the plains of Africa hunting antelope or whatever the hell it is. Number three, infection. Clearly, viruses can push it over the edge as well, or bacteria. Now, the great breakthrough, in my view, in autoimmune disease is EBV and MS, which you may have seen. I mean, that's a really exciting development. We always suspected there was a viral origin anyway. And who would have thought of EBV, that we're all infected with? So we all carry EBV, remember, because it's one of the commonest things. So it must be EBV when you're in your teens, probably activating an inappropriate immune scenario because of genetics that's in there as well, going latent, and then that disease begins to emerge. The simple pathogenic mechanism is epitope mimicry, because it's got proteins that look a bit like myelin, and maybe that would kick things off. So I think that discovery means we're getting closer now to an origin for an autoimmune disease, this kind of thing. So I think that's part of the mix as well.
All right, so maybe changing gears a little bit. I mentioned it at the beginning that you also are very keen into science, kind of communication, and you actually have a podcast as well.
I do.
So maybe do you want to share that a little bit with us? How is that? How did you end up doing this, and how's your experience? Broadcasting on public Irish radio.
Yeah, well, you see, it's a good question. Sometimes many people don't like this. Remember, what's that guy doing, banging on about things on the radio? So often people are reluctant to do the communication thing, and I'm a big advocate for it. I think it's essential that we do communicate science to the public. There's a few reasons for this. One is they're paying for our grants often. They're taxpayers, and the money no longer in the US, sadly, Jason, but at least in Europe, the taxpayer is still funding science, which is great. So we have an obligation to tell them what we're doing, Mm-hmm. Secondly, massive appetite out there for this, remember. Thirdly, there's misinformation the whole time as well. Yeah. So those are the reasons. And it can be fun, more than this podcast, It can be a bit of fun. So those are some of the motivators for me. But I came into it stepwise. It began, I'd say about 15 years ago. in my university, we had a special outreach program for the public. It was called the Science Gallery. I got involved in that, giving the odd lecture to the public. I quite liked it. Thinking, oh, this is quite nice, And next thing then, I'll be on the radio occasionally. The Irish radio would ask me mainly to talk about vaccines or immunology, or something I know something about, mercifully. Then I got a regular slot on the radio every week, and I began to talk about other science topics that interested me. So it kind of was ramped up over the course of 10 years or so. And then now and during the pandemic, of course, it was amazing because they asked me to go on the whole time, talk about, COVID and all the usual things. And I could talk about it because it is in our business, Yeah. And my lab began working on COVID. We began doing stuff on therapeutics. We published a couple of papers on coagulation and the coagulopathy in COVID, for instance. So I was able to speak on it with some authority, you might say. And then secondly, I was an advocate for vaccines, being an immunologist, of course, And then since then, that's when I was at its most intense, shall we say. And then the podcast thing came up, and I began doing my Show Me the Science podcast. So I guess the best answer is it was a stepwise thing. I began being a tiny bit annoying. Now I'm very annoying. Let's put it that way.
Very entertaining, I would say. Annoying is...
Well, you see, the other thing that motivated me, Brenda, was when I became an academic, which is about 30 odd years ago, they gave me the first year biology class to teach. Isn't that amazing?
Yeah.
Now, can you imagine a bunch of 18-year-olds with no reason? They were interested in biology. They're very distracted. How would I engage them? And that was like talking to a general audience. And I began talking about, mitochondria or whatever, and had to learn then how to communicate to a bunch of 18-year-olds. It's no different to that. And I've never seen it being any different to that, actually. So that got me into it, I guess, from my teaching. And that's why teaching can be very useful. I think I'm a big advocate for anybody who's listening. Always do a bit of teaching, even though you might like it, because you learn how to communicate. If you teach about what you're working on in your research, it actually gives you ideas incredibly as well. Secondly, tell the public what you do if you can. It's not for everybody. Some people don't like doing it. Some find it awkward. That's fine. That it's not compulsory, But if you have a little hankering towards it, start doing it, and you might actually enjoy it. And then the other thing is, you get a huge positivity. People love it. They say, "Oh, thanks for telling me that." I get emails. I heard your podcast on whatever. I did one recently. There was a great Nature paper on after a heart attack, monocytes go in and repair the heart. Sleep promotes the monocytes to go in, this kind of thing. Oh, that was very interesting. My dad just had a heart attack. I could talk about this to him. So it is very rewarding as well. And I'm sure you guys, I hope you get feedback on this podcast, because it's a rewarding thing to do.
It is. I love it when we hear back from a listener. It's amazing. So
we've had
more
listeners lately, haven't we? We've had more listener info coming in, so we really appreciate that.
Absolutely.
It's a bonus, Jason. Most people wouldn't be emailing me now. In the pandemic, I had a deluge of emails, of course. But most of the time, you don't get even one bit of feedback. There's probably 10 people think the same way, you see. So.
Yeah. All right. Well, we're starting to run out of time, but we always end with an interesting non-science question. Yeah. So, if you could have any superpower, what would it be and why?
To get grant money without writing grants.
So independent wealth. The Bruce Wayne power.
The Bruce Wayne. Oh, good Lord, that's a very challenging question, Jason. I think
independently wealthy. You're just
that's a superpower these days.
To become invisible. If I could click my fingers and suddenly become invisible, that'd be great, wouldn't it? Because you could sit in those grant committee discussions about your grandmother dissing you. You sit there and hear the truth. So that could be useful.
That seems like a good. I was thinking of, you can also just go and take the money from a bank to finance your research, but I guess also
if you're invisible, now we're talking about you could go and rob a bank, and now we can't be advocating that.
Getting the gossip is also pretty cool.
Independent wealth is now the new superpower, as we've all learned.
Yes, you can become president of the United States with it. Apparently,
let's not mention the Musk name. Yeah, I mean, look at it.
President 1 and President 2. Yeah, I know.
Yeah.
Guys, look, like being an American right now is a wee bit difficult.
It is. It's certainly interesting, Jason. I listen, as I said earlier, you guys will come back. Don't worry. This storm will pass,
Yeah.
Once the right people do the right thing, you'll be all right.
Yeah. Just a few years in the wilderness. It'll be okay. I hope. All right. Well, thank you for coming on. This was an absolute blast.
Absolutely. Yeah. Thank you so much. We had Professor Luke O'Neill from Trinity College Dublin joining us to the podcast today. Awesome conversation. Thank you for being with us today.
Thank you very much, Brenda and Jason. That was great fun. Thanks.
Well, after this great conversation, we are reaching the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get show notes, including episode summaries and links to all of the interview and Roundup Papers. And you can also reach out to us on X at Immunology Podcast or via email at info@immunologypodcast.com if you had feedback or you would like to suggest a guest or a paper, just say hi. We're always happy to read your emails. And we're gonna hear us next time in our 100th episode, which is we're very happy.